Hey everyone, my name is Nick Porch. Technically I'm a geologist, let's say, and I'm an honorary bug person for today, let's say that. And I'm going to be talking about assessing the impacts on narrow range endemic beetles. And I'm talking from Wurundjeri country, and I pay my respects to the traditional owners and from the land of which I present today. I'm going to be talking about what we think of as narrow range or short range endemic species. And this sort of stemmed from the fact that one of my colleagues came to me and said, basically, we should come up with a project. What are you interested in? And this was an ecological project rather than perhaps a project like I would normally be interested in, which is much more in line with human impact on biotas and biogeography in particular. And it really stems from an interest I have in a group of Treckine beetles that are little small ground beetles that are found in wet forests. And many of these species are widespread throughout southeastern Australia. And kind of interestingly, many of them occur in regions that were fairly heavily impacted by fire. So the one that really interested me in particular was the one up at the top here, Eutrachops ovalis. And it was because the area that it occurs, it was very heavily impacted by fire, but no one had mentioned it. And this was something that had come across fairly often. And in this sense, I was thinking, is it really about what we don't know or what we do know? And how do we start to figure out what we need to know? In this context, the Great Dividing Range of Eastern Australia is what we might think of as a mesic archipelago. For wet-adapted biota. It's reflecting the fact that Miocene-Pliocene drying has driven large numbers of taxa into an upland archipelago where they've speciated extensively, topographically delineated. And this happens across many groups. And Mike Ricks was talking about the spiders in the previous session. One of the problems is that much of this fauna is relatively poorly sampled at high geographic resolution. And that means that online data frequently poorly represents both the diversity, but also the geography of where these taxa tend to occur. Many of these short-range endemic species are not represented in Atlas of Living Australia. And it's not only true of small things, this is also true of large things. So one of the taxa that we found when we were doing this particular project is we came across new species of things like Lesotes stag beetle. So these are things that people are really into in terms of collecting or in terms of understanding the distribution and the like. So we don't only not have knowledge about the small stuff, through this sort of mesic archipelago, there's lots of new things we can find out. So just thinking about a whole range of different taxa, coming at the literature and looking at what's known, here is the genus Nargiotes. On the right-hand side in the inset, I have ALA data, and in the main map here is the distribution of all the species. And each of the narrow range or short range endemic species has a little ellipse around it. Paraptictus, again, hardly anything in ALA, but lots of diversity, narrow range endemism. Similarly, when we start to get to groups like Tympanagaster, which are little hygropetric beetles that live on waterfalls, massive amounts of narrow range endemism, and lots of species along this throughout this region, but almost nothing in ALA. And similarly, when we get to things like little blind carabids. So what does this mean? Well, what it means is when you start to add it all up together and look at the distribution, on particularly the geography of the speciation or the geography of places that have lots of short-range endemics, then you start to see really clear patterns that exist throughout this region. And this is perhaps not that surprising. Given this was an ecological project, one of the ways that we went about this was to do a paired project where we looked at burnt and unburnt sites, 20 on the escarpment and 20 subalpine. I'll show you those in a moment. And we also divided each of those into three different sort of what we might think of as microhabitats. Ones are rich in logs, ones that are very rocky, and ones that are much wetter along creeks and the like, each with 3 replicates. I had 5 pitfall arrays with 3 tubes. The 3 tubes were combined up into one sample, meaning that we ended up with 3,600 pitfall samples, which is a fairly significant number. This is a distribution of sites, largely sort of thrown down to sort of pick places that we knew had high levels of narrow range, short range endemism, and places that sort of fell between them to test for whether that short-range endemism is much more highly geographically distributed than we have the current data for. So one of the problems, of course, is we're dealing with a poorly known fauna, and we're doing this in COVID with no museum access. So we had a whole range of issues that have meant that what we're dealing with and what we're talking about largely today is really sort of, we might think of as preliminary, and that's something that's a theme that's running through multiple talks today. So each of these groups was sorted to morphospecies, many are identified to genus. Every morphospecies was imaged, often multiple times. So at the moment, I have 5,000 images of these things. They were counted and recorded by sample. And now with museum access back on, we're going to be able to at least increase the quality of the data that we have somewhat. That's perhaps not surprising. So what did we find? Well, we got over 45,000, almost 46,000 beetles, mostly from pitfall traps, and lots of other little things, including many that are really poorly known. So the one on the top right-hand side there is a springtail genus called Acanthanura, for example. There are known mainland species described in this genus. We got 223 of them, and there's at least 8+ species there. So a lot of diversity, and in the beetles, but in everything else that we got, a lot of unknown diversity in many ways, or unrecognized diversity. In the beetles, we got 1,554 taxa, and some of them were only sorted to group, for example. So things like staphylinids, which Chris Reed alluded to before as being difficult. Some of the staphylinid groups, like Aleocorines, we didn't even bother with. It's way too hard. So we ended up with about 1,500 morphotaxa. Now, not surprisingly, everything responds differently. So there's groups that respond really happily to fire, like Anorthorhinus and Microchetes on the left-hand side here. Microchetes eats moss, and I suspect that Anorthorhinus might also eat moss, so it's not surprisingly that they love fire, because mossy habitat is what you see straight after the fire. There's a whole pile of stuff that really doesn't care. So we see even in a Gondwanan taxon like Guthaloma rotundulum, it really doesn't care whether it's burnt or not. And then of course, there's a whole pile of stuff that really doesn't like fire very much at all. So things like Eurylichnus blackgravii, which is another Gondwanan carabid or ground beetle, for example, it really didn't like the fire very much at all. And similarly, something like Mandelatina, this is a composite, this is a sort of undescribed genus probably, and in it there's lots and lots of narrow range endemic species. So this data is really just reflecting the fact that we've lumped all of them together to look at whether there's patterns, and they really don't like the fire as well. Just like Chris showed, it's not completely shitty for dung beetles. That's really nice to see in some ways. So the one on the Mathusius on the left, Mathusius illawarrensis was one of the species that was on the priority list. Now remember, this is not dung baited traps, this is unbaited pitfall traps, so the numbers of individuals are quite low, but it doesn't really care whether it's burnt or not. And both Lepunus, which is a composite of 3 or 4 different species, and also a new species of Thyregis, which is found in the south of the region, even though there's very, very few records for it, none of these are significant. So the numbers of individuals decline, but they're not really significant in terms of whether it's burnt or unburnt. What I should say is something about what the ecologists did. And one of the major phenomenon that we saw is for species that we considered short range endemics in the scarp sites, they significantly declined in abundance as fire severity increased. And that was one of the major patterns. Remember here, we're not looking at individual species, we're just looking at whether they are short range endemic species or not. And in the ecological data, where we're looking at microhabitats, there was only one real significant difference in terms of short range endemic species, and that was sites in the mountains with logs had more than the other sites. So that's perhaps not that surprising. One of the really interesting things to do was to run something like some analyses where we looked at things like how similar the sites or the assemblages at each of the different sites are. And this is just a hierarchical cluster based on cube root data. So we're downplaying the impact that abundance has. And what it really shows is it divides up into two different bunches, the scarp sites and the montane sites. They're ecologically quite different. And within them, there's significant bunches of sites that tend to cluster together. And when we have a look at them on a map, we can see that they're basically clustering in certain regions where we have some idea that there should be endemism. Now, this is just assemblage data. A lot of the short-range endemics, for example, aren't hugely abundant, which means that they're probably not, in many cases, contributing to these types of patterns where we're analysing data in this type of way. But we can see, for example, that the Snowy Mountains in the middll, Errinundra on the bottom in far eastern Victoria, and a number of the escarp sites divide up fairly significantly, just in terms of the total sort of assemblage-based data that we have. Now, just to show you that we have dealt with individual narrow-range endemic taxa, here's something rather beautiful. This is a Genuacheles species. Genuacheles, I call this one lentil. It looks just like a little lentil, and when you look at it from the front, it's just this really weird little lentil-y looking thing. And this is the distribution of Genuacheles species. This is a group that in the Australian region has 3 described species, and there's hundreds of undescribed species known. And it's not surprisingly then, when you start to look at the distribution of these Genuacheles species throughout this region, and they're dominantly in the scarp sites, then there's lots of turnover spatially throughout this region. And what it's showing us is that there's actually a lot more detailed, or a lot more fine-grained resolution in terms of the short-range endemic species than we might imagine if we start to look at what we have known from, for example, the literature that I quickly went through for those first 4 groups of taxa. Interestingly, just this little bunch down here, this is the bigger valley where we have the little, the circle, for example. And if we were to take lots of other genera, like similar cryptorhynchine weevils, Rhoptoporus, Pseudomydas, and a whole series of them, that little area has repeated patterns of narrow range endemism, and many of these areas have repeated patterns. So once we get our data better sorted out, we'll be able to better understand what's going on. So I guess what this has sort of got me to think about is a whole range of questions that we need to sort of begin to think about and resolve. And this, as a group of people interested in biodiversity, particularly invertebrate biodiversity, and interested in space, then there's a whole range of things that we need to think about. How do we deal with undescribed species that we know? A lot of these species are in collections. We know where they live, we know what they do, but it's really difficult in that sense to deal with them. How do we convince managers that these little things matter? Then I think one of the ways is which we can start to do this is to start to provide some of this data and on the patterns of geography rather than on necessarily the systematics or the taxonomy. And have we forgotten about stuff like literature? I mean, why does everything have to be in the Atlas of Living Australia for it to be meaningful in terms of telling us about patterns, telling us about diversity? And I guess the last thing to think about is we just basically need more people on the ground, right? Doing a lot of this work, but how do we get that? And I guess what the final sort of thing to say is that a lot of these faunas have been much more resilient than we've thought about. So that when I initially started thinking about Eutrechopsis ovalis, we found multiple specimens from multiple sites, but always in those little wet habitats. And this, of course, this type of research involves lots and lots of people, and it's been a rather large undertaking that seems like it's never going to finish. So thank you for listening for that really quick overview, and I'm happy to take questions if we have time.
Thanks, Nick. That was excellent and massive amount of work and very valuable as well. So does anybody have any questions for Nick? So I've got a quick question, Nick, just in terms of defining short-range endemics, what criteria did you use to define short-range endemism? I've had to do a similar thing and it can be tricky. What did you use to? Yeah,
well, basically, we just took, I think, Mark Harvey's paper where he says short-range endemics are things that are less than 10,000 square kilometres, which is incredibly massive because almost all of these things have incredibly short ranges. I just had an honours student who's finishing up working on a species in the Dandenong Ranges, which is a short range endemic. It has a 4 kilometre wide range. So if you make it 4 kilometres by 4 kilometres, that's 16 square kilometres. We really need a term which actually, I think, extreme short range endemics or something like that. I mean, because in invertebrates, I think we have a lot of extreme short range endemism.
Yeah, no, I agree. Yep.
Yes, can I ask one quick question of Nick?
Go for it.
Do you think the larval stages of these insects are going to hold more important ecological information than the adult stages?
Don't ask me about larvae, Mark, because beetle people know nothing.
I just did.
I know. Beetle people know, well, beetle people like me know nothing about larvae. I mean, we don't even bother with larvae. None of the, I mean, of course, we haven't even included larvae in those numbers I just showed you. That was adults, of course, largely because we can't actually relate, unless we go down the genetic path, which is a whole different program, of course. We can't actually relate what's going on, what larvae and which adults match, of course, as you can well and truly probably imagine.
Yeah.
It's tricky.
Thank you.
Excellent. Thanks very much.